Welcome to the Friday evening lecture. We'll begin in just a moment.
Good evening and welcome to tonight's Friday evening lecture. I'm Nipam Patel, Director of the Marine Biological Laboratory. The MBL is dedicated to fundamental biological discoveries that inform human health and the health of our planet. Every summer since its founding in 1888, the MBL has hosted public evening lectures where renowned scientists, historians, and architects of modern biology have presented stunning scientific discoveries for exploration and discussion. Despite the hardships of the war years and the Great Depression, the Friday evening lectures have always been held without interruption. Just as our predecessors did, we are coming together as a community to adapt this year's series with streamed lectures delivered by notable Friday evening lecture alumni. While I regret that we can't be together in person in the Lilly Auditorium, your presence here tonight is helping us to keep this important tradition alive. Thank you for joining us and enjoy the presentation. And remember to turn off your cell phones.
I'm very happy to be introducing Bob Langer for this Friday evening lecture at the Marine Biological Laboratory. Robert S. Langer is one of 12 institute professors at MIT, which is the highest honor that MIT awards to its own faculty members. He is widely recognized and highly cited in the fields of bioengineering and biotechnology, especially in controlled release drug delivery and tissue engineering. His publications have been cited over 300,000 times, and his H-index is a phenomenal 272, which makes him, according to Google Scholar, one of the top 10 most cited authors in the history of science. Bob also holds over 1,300 patents. His laboratory at MIT is the largest single PI-led laboratory in biomedical engineering in the world. Bob Langer was born August 29th, 1948 in Albany, New York. He received his bachelor's degree from Cornell University in chemical engineering and his SCD degree in chemical engineering from MIT in 1974. From 1974 to 1977, Bob worked as a postdoctoral fellow for cancer researcher Uta Folkman at the Children's Hospital in Boston at Harvard Medical School. Bob is widely regarded for his many contributions to medicine and biotechnology. He's considered a pioneer of many new technologies, especially including controlled release systems and transdermal delivery systems, which allow the administration of drugs or extraction of analytes from the body through the skin without needles or other invasive methods. Langer worked with Uta Folkman at Boston Children's Hospital to isolate the first angiogenesis inhibitor, a macromolecule to block the spread of blood vessels in tumors. Macromolecules tend to be broken down by digestion and blocked by body tissues if they're injected or inhaled, so finding a delivery method for them is difficult. Bob's original idea was to encapsulate the angiogenesis inhibitor in a non-inflammatory synthetic polymer system that could be implanted in the tumor and control the release of the inhibitor. He eventually invented polymer systems that would work for this purpose. This discovery is considered to lay the foundation for much of today's drug delivery technology. He also worked with Henry Brem of the Johns Hopkins University Medical School on drug delivery systems for the treatment of brain tumors. The wafers or chips that he and his team have designed have become increasingly more sophisticated and now can deliver multiple drugs and respond to stimuli. Langer is regarded as the founder of tissue engineering in regenerative medicine. He and researchers in his lab have made advances in tissue engineering, such as the creation of engineered blood vessels and other types of engineered tissue. Bioengineered synthetic polymers provide a scaffolding on which new skin, muscle, bone, and entire organs can be grown. With such a substrate in place, victims of serious accidents or birth defects can more easily grow missing tissue. Such polymers can be biocompatible and biodegradable. Bob Langer is also involved in several projects related to diabetes, working to produce an implantable device to treat type 1 diabetes by shielding insulin-producing cells from immune system attacks. I'm both proud and humbled to say that I have known and considered Bob a friend for the last 40 years, since the late 1970s. One personal memory that has stuck with me is that I believe Bob was the first person that I knew who had a mobile phone. I remember his using one in the late 1980s when we were both on the External Advisory Committee of Chemical Engineering at Georgia Tech. This early adoption of technology has led to one of Bob's most endearing personal characteristics. If you email or text Bob saying you would like to speak with him, you will generally hear back with him within minutes, not hours or days. This speediness is only one manifestation of the generosity that accompanies Bob Langer's technical brilliance and unmatched record of accomplishment. His lab has spawned many, many highly successful professional progeny. I'm happy now to turn the virtual stage at MBL over to Bob Langer for the MBL Friday night lecture.
Well, it's a pleasure to speak to everyone tonight. My background is I'm a chemical engineer. I actually got my doctorate at MIT in 1974. And when I finished my degree, almost every one of my friends went to work in the oil industry. But I wasn't very excited about that. And even though the jobs were very high paying, I actually had this idea, because I helped start a school for poor kids in Cambridge, that I could do something in education. And I had done a lot of work on developing new chemistry curricula when I was at the school. And one day I saw an ad to be an assistant professor of chemistry education at City College of New York. I was very excited about that. I wrote them a letter applying for a job, but they didn't write me back. But I really liked that idea, so I found all the ads I could to do something like that. I found about 40 of them. I wrote to all these colleges to see if I could be an assistant professor to develop new chemistry curriculum, and none of the 40 wrote me back. So I wasn't doing very well on that. Started to think, what else might I be able to do that could help people? And one of the things I thought about was medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write back either. But then one day, one of the people in my lab, Barry Bunow, he said to me, he said, Bob, he said, there's a surgeon in Boston named Judah Folkman. And he said, sometimes he hires unusual people. So I wrote to Dr. Folkman, and he was kind enough to offer me a job. And on the first slide, I'm going to show you what his idea was. If we could put the first slide on, please. So Dr. Folkman had this idea that if you could stop blood vessels, that might be a whole new way of stopping cancer, because you would cut off their feed, basically, and their nutrition. So and people were very skeptical about that. So one of the things that we talked about was, well, where could you ever find something that might stop blood vessels from growing? And we thought about cartilage. And so I got some little rabbits from the lab. I got some of their cartilage, but I couldn't get very much. But one of the things you learn in chemical engineering is scale up. So I went to a slaughterhouse in Cambridge, and I got a cow bone, but still one bone I couldn't do that much with. So I made an arrangement with these meat packing places in South Boston to get all their bones, and I brought them back to the lab, and I scraped the meat off of them. And on the next slide, I'm just going to show you a picture of one of them. If we could go to the next slide, yeah. And the top part is the cartilage, and I scraped everything off of it, and then I'd slice it off, and I'd slice thousands of these off, and I'd put them and I'd extract them with certain chemicals, and then I would wanted to test them. Now, that brings up a second problem. One of the biggest issues when you're trying to isolate a new substance or set of substances, having a bioassay. And there was no bioassay to study blood vessels at the time. So we had to invent one. And on the next slide, I'll show you what Dr. Folkman and I started talking about, was that maybe we could take certain tumors like V2 carcinomas, which will, if you put them in an eye, cause blood vessels to grow. And then what we wanted was a controlled release polymer. This was 1974, a controlled release polymer that could deliver whatever we were isolating. And of course, most of the things that were in cartilage were large molecules, like proteins or peptides or other things like that. And so we had to be able to have this polymer do two things. One, not cause irritation in the body, which is hard, but two, be able to deliver large molecules. So Dr. Folkman asked different people. He was on the board of a company called Alza, which was really the only place in the world that was doing any work even remotely connected to this. Could they help us? But they all said you couldn't release large molecules from polymers or solid materials. They said, they were just too big to go through. It would be kind of like any of us walking through a wall. And in fact, the literature said the same thing. The next slide's an example of that the use of polymer matrices for slow release systems has been virtually restricted to small molecules. I think the only thing I had going for me is I hadn't read the literature. So I spent about 2 years working in a laboratory, experimenting kind of almost Edisonian-like with different techniques. And I found over 200 different ways to get this to not work. But finally, I found one way, next slide, to get it to work. And I was able to make these tiny particles, microparticles or nanoparticles. You see a picture of one and one cut in half. And we were able to show using this that for the first time, this is shown in the next slide. This is a paper Dr. Folkman and I wrote in Nature in 1976, that you could release molecules of any size, really, for over 100 days. And we even showed for the first time that you could release nucleic acids like DNA or RNA. And so that was very exciting to us. We also showed that you could get constant release. Later on, this was work I'd done at MIT, where myself and some of my students worked out ways to do mathematical models so you get constant release. And a picture of that is shown in the next slide. The thing is that when I first presented this work at scientific meetings, it met with a tremendous amount of skepticism. I remember in 1976, I gave a talk for the first time at a big scientific meeting. It was in Midland, Michigan. It was a big polymer meeting. And I remember being incredibly nervous and giving this 20-minute talk. And at the end of the talk, I thought I did all right because I didn't forget too much of what I was going to say, or I didn't stammer too much. So I thought when I was done, all these older scientists, chemists and engineers at the meeting would want to encourage me because I was this young guy. But when I got done, a whole bunch of them came up to me and they said, we don't believe anything you just said. They said, next slide, that just like the literature, that the large molecules can't slowly diffuse through solid polymers. And they also said that the solvents we were using would destroy whatever we put in. And actually, things kind of went downhill from there. I tried to write grants, and my first 9 grants were rejected pretty soundly. People said, like, how could a chemical engineer do cancer research or anything like that? And then I was a postdoc, as I mentioned, but, a lot of and I liked that, but a lot of my friends said, it's probably not good to do that forever. So I started applying for faculty positions. And as I mentioned, I was a chemical engineer, so I applied to chemical engineering departments. And no chemical engineering department in the world would hire me. They said, this bio stuff you're doing doesn't make any sense. That chemical engineers don't do stuff like that. So I didn't get a job in a chemical engineering department. I ended up getting one in a nutrition department at MIT. But and the reason I got that was because of the head of the department, Nevin Scrimshaw. He was what I'll call kind of a benevolent dictator. He was a visionary guy, and he liked me, so he hired me. But he didn't bother to ask the rest of the department what they thought. So, and that probably still would have been okay, except for the fact that the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. In fact, the next slide is an example of that. This is from Michael Marletta, who was one of my very good friends and a colleague of mine at MIT at the time. Now he's got an endowed chair at Berkeley. He's a member of the National Academy of Sciences. And he was saying that this is referring back to 1980. He was giving this at an award thing I got. He said, well, one evening I went to a faculty dinner at a Chinese restaurant with Bob Langer and some senior MIT professors. He said a senior scientist sat quizzing us while smoking a cigar. He said when the older scientists heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. And Mike said he thought he was in a Fellini movie. But anyhow, I kept at it, and people began to use these techniques. And then we started to understand, well, why did this even work? And to understand it, what we do is Rajan Bawa, one of my students, we'd take a cryomicrotome. It's a low temperature cutting instrument. We'd cut thin sections through the polymer. And the next slide shows an example of that when there's no drug in it. And the slide after that shows what happens if we put a reddish drug, myoglobin, in it, and you get, this is before any release, and you get what we call a phase separation, where the drug is white, I'm sorry, the polymer is white and the drug is red. And it turns out if you release these for a year and then you cut a thin section, what you see is left behind where the drug was are these pores. That's on the next slide. And these pores, it turns out, are large enough so that molecules any size, even DNA or RNA, will go through it. But the way you can control release rate, it turns out, is these pores are interconnected and they're very winding and tortuous. So it takes a really long time for the molecules to get through it. One way I sometimes explain it to people is it's kind of like driving a car through Boston. Boston and these systems have what we call a very high tortuosity. That's a chemical engineering term. So anyhow, over the years, our students have done models, and now we can, get a release anywhere from almost instantaneously to many, many years. And because we could do that, we could now study the angiogenesis problem. And the next slide shows a picture of that. And what you see is the assay. The tumor is there and the polymer's there. And we actually did several thousand eyes. We tested over 100 fractions, and almost every one of them didn't work. I'm going to show you the one that did. We actually repeated this 21 times, but this is kind of graphic, so if people don't want to look at blood, you shouldn't look. But what you'll see on the next slide is a rabbit assay. This is from a paper Dr. Folkman and I and others wrote in Science in 1976. But what you see in the one panel is the sheet of blood vessels. This is 9 weeks after the start of the experiment, growing over the polymer on the way to the tumor. In fact, 3 weeks after this, the tumor would be three-dimensional. It'd be out of the orbit of the eye. It would actually kill. We sacrificed the animals well before then. But on the other side, what you see is the tumor and the polymer. But notice that the blood vessels are much sparser. They're lower, and they actually avoid the polymer completely. So we repeated this 21 times. It did the same thing. And actually 60% of the time, these tumors never took off. And that never happened in any of the controls, even though there were thousands. So this really showed for the first time that you could now graphically see that blood vessels could be inhibited, really changed conventional wisdom. And nonetheless, it still took another 28 years and literally billions of dollars of research by different companies like Genentech before the first clinically approved angiogenesis inhibitors were on the market. So this is 1976. On the next slide, I'm going to show you a table, which may be hard to see. But what happened starting in 2004 is one after the next angiogenesis inhibitor was approved by FDA. So Avastin by Genentech, that's been one of the biggest, maybe the number 2 or 3 best-selling biotech drug in history. And this keeps going. Now they're used for many different types of cancers. They're used for really the only treatments for diseases of blindness in the back of the eye. And these are drugs like Eylea or Lucentis. So these are, like I say, used by many millions of people every year. And so this was exciting to us that finally, after many years, we'd see all these making a big impact on patients' lives. We also thought that the drug delivery work we did also could be useful in its own right. Larry Brown, one of my graduate students at the time, showed for his thesis, next slide, that you could take insulin, put it in a little pellet, and that you could put in a diabetic rat, and it would lower the blood sugar for over 100 days. And Dr. Folkman said to me one day, said, Bob, we should file a patent. And that might seem common today because, we have a lot of patents, but then we had none, and Boston Children's Hospital actually didn't have any either. So we filed for the first patent in the history of the hospital, and 5 years in a row, the patent examiner rejected it. And so the head of the licensing office at the hospital told me we should just forget it. But I don't like to forget things like that. So I started to think, how could we convince the patent examiner to allow this patent, legally, of course. And as I mentioned to you in the beginning, when we first did this, everybody said it was impossible. It never worked. So I wondered, did anybody ever write that down? So I did what's called a science citation search. I did this in 1982, and looking back at our 1976 Nature paper to see who wrote about it. And I found a number of useful things. I'll just read you one quote, next slide, from 5 of the top polymer chemists in the world. And what they wrote was, generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates through polymers. However, Folkman and myself have reported some surprising is a really good word for a patent examiner, some surprising results that clearly demonstrate the opposite. So I showed this to the hospital people, and they went down into Washington to show it to the examiner. And he said, gee, I had no idea. He said, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from each of these 5 people that they really wrote this. So I was this young guy, I wrote them, and they were nice enough to write me back that they really wrote it. And we got this really broad patent shown on the next slide. And with that, I got involved in starting companies and we licensed it to different companies. We taught different companies. And today, based on this, there are many, many products on the market using these kinds of technologies. The next slide shows a picture of a number of them. I won't go through all of them, but there are many different ones. Some are useful, like 6-month systems for preventing prostate cancer or endometriosis. There's many of the opioid addiction systems are based on these, and various treatments for eye disease, for stopping bleeding, for treating schizophrenia, and many others. So these are just a few pictures. So now many of these systems are on, being used by many millions of people to treat a whole range of different diseases. I also thought that we might even be able to do better for some cases and even create nanoparticles. In the next slide, that you could inject into the bloodstream. But one of the challenges with nanoparticles is that they may be taken up by cells you don't want, like macrophages. So we developed a way to make nanoparticles where we took something like polyethylene glycol, that's a substance that's safe by the FDA, and coat it on these particles. And when you do that, it actually will avoid the immune system. And then what you could even do, next slide, is actually put a targeting molecule on. Omid Fargadazad, when he worked with us, did a lot of that. And I'll just show you a video on the next slide that Nova did when they were, the TV show, when they were in our laboratory discussing nanotechnology with me.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
I should say that a lot of the clinicians I work with tell me it doesn't blow the cell up that way exactly. But anyhow, this kind of technology is now being used for all kinds of things, with polymers, with lipids, to do different things. In fact, Alnylam, a company that I've done a lot of work with, has several FDA approvals using using nanoparticles to deliver siRNA. Moderna, a company that I started, is using nanoparticles to deliver the COVID vaccine. It's absolutely critical to have these nanoparticles to protect these molecules like siRNA or messenger RNA, and then to deliver them to the cells you want. Now, a whole other thing that we did was actually look at materials. I was curious about how materials found their way to be used in people. And I wondered whether it was done by chemical engineers like myself or other people. And what I found was almost always the way materials found their way into medicine is that a medical doctor urgently wanted to solve a medical problem, and they'd go to their house and they'd they find a material that kind of resembled the organ or tissue they want to use. And the next slide illustrates this. And I'll just give an example, the artificial heart. Back in 1967, some of the clinicians want to make a heart that, and they asked, well, what object kind of resembled the organ or tissue that you want to make? And they said, well, a lady's girdle, because it has a good flex life. So they took the material in the lady's girdle, and that's what they made the artificial heart out of. But the problem with that is that, as you might read in the newspapers, is that when blood hits the artificial heart, the lady's girdle, it can form a clot that could go to the patient's brain. They get a stroke and they die. But if you think about it, something that was designed to be a lady's girdle, may not be the optimal blood contacting material. And this problem really pervades all of medicine. Like, for example, breast implants. One of them was a mattress stuffing. So I thought, being a chemical engineer, you learn about design. And I thought, well, maybe rather than take these materials from your house, what if we could synthesize them from scratch and ask the question, what do we really want in a biomaterial from a chemistry standpoint, biology standpoint, and engineering standpoint, and then design them for first principles. So we picked an example. The next slide it will illustrate that. When we started in 1980 on this, the only material that had been approved by FDA that was synthetic and degradable displayed what we call bulk erosion, meaning it starts out like that sheet on the top, but then it gets spongy and then it falls apart. And that might be okay for some drugs, but for others, it could lead to a burst, like insulin or an anti-cancer drug. You could get bursts, and that could be fatal for a potent drug. So we said, well, what do you really want in a biomaterial? And we said, well, you want the next slide, which is more what we call surface erosion. Then that can't happen. So we said, well, how could we make a material that does that? And I won't go through all the chemistry, but we'd ask different questions like, what should cause the degradation? Should it be water? Should it be enzymes, something else? And we actually wrote on the blackboard, Bob Linhart and Howie Rosen and others, some of my students, exactly how we want to go about that. And we came up with a chemical formula that we drew. The next slide shows that has anhydride bonds connected to it. And then these units, I promise I will not show, and try not to show more chemical formulas. But basically, this was the idea that we had, was that this would have the right degree of what we call hydrophobicity to repel water and would still degrade at the right rate. And we did a lot of work to prove that. On the next slide, this is a test tube experiment, but we showed when we changed the ratio of those two units from 79% to 55% to 15%, we could make these dissolve at almost any rate we wanted. So what happened is that we could make these last anywhere from 2 weeks with 79% to many years with the 0%. And one of the things that Henry Brem, who was one of my colleagues at Children's Hospital and then became young professor, a neurosurgeon at Johns Hopkins, asked me one day, he said, well, could we use this to come up with a new treatment for glioblastoma multiforme? And some statistics he showed were on the next slide. It's uniformly fatal. And he was looking for a better way to deliver BCNU. That's shown on the next slide. And this is a very toxic drug. But the idea that he and I talked about is shown on the next slide. And that is, we're going to allow the neurosurgeon like Dr. Brem to line the surgical cavity with little wafers that contain this drug. Now, normally this drug only lasts for 12 minutes, but if it's put in the polymer protects it from degradation. And the idea is Henry would put, or other neurosurgeons would put it in they do the surgery, they take as much of the tumor out as they could, but then they'd leave this behind, some of these behind before they close the patient up. And so you'd get high concentrations in the brain where you want to, and low concentrations in the rest of the body where it might cause harm. Now, whenever you do this kind of work, you have to raise money. And so the way we would raise money is we would write grants to like the NIH or other places, and then they'd have study sections review it to say what they thought. And we didn't do very well. I actually saved a lot of the grants, grant reviews. The next slide just shows some of the responses. So we first had the idea about synthesizing these new polymers, those chemical structures I showed you. A lot of the chemists in the review section said, well, you can't synthesize them. It's not possible. But I had a very good graduate student, Howie Rosen. Howie later became senior vice president of Gilead and actually president of ALSA, the company I mentioned, and has been elected to the National Academy of Engineering, and he synthesized them. So we sent the grant back, and the reviewers then said, well, okay, that's good, but these polymers, they have anhydride bonds, they'll react with whatever drug you put in. But I had another couple of very good postdocs. Cam Leung, who is a chaired professor at Columbia University, also was elected to the National Academy of Engineering, and Bob Linhart at RPI, who's elected to the National Academy of Inventors, and they showed that there was no reaction. So we sent it back again, and the reviewer said, well, that's good, but we still shouldn't fund it because the polymers are fragile. They're low molecular weight, they'll break. So, but I had another couple of postdocs, Avi Dome, who later became head of medicinal chemistry and pharmacy at Hebrew University, and Edith Mathiowicz, who's a full professor at Brown and a member of the National Academy of Inventors, and they actually made polymers high molecular weight, and they were very strong. So we sent it back again, and the reviewer said, well, okay, but these polymers have never been used on people. They're certainly going to be toxic, so we still shouldn't fund it. But I had another graduate student, Cato Lorenzen. Cato later would become Dean of Medicine at University of Connecticut and actually received the National Medal of Technology from President Obama. He showed actually they were very safe. And then, so we sent it back again, and the reviewer said, but the drug won't diffuse far enough to kill remaining tumor. But I had another graduate student, Mark Saltzman, and Mark actually became the first head of biomedical engineering department at Yale, been elected to both the National Academy of Medicine and the National Academy of Engineering, and he showed that it did diffuse far enough. Anyway, this kept going on until 1996 when the FDA approved it, which was the first time in over 20 years they approved a new treatment for brain cancer, and the first time they ever approved any treatment for local chemotherapy. You can probably tell from the way I'm speaking that I'm very proud of how well all of our graduate students have done. They became, heads of departments and schools and received all kind of honors, where the reviewers, that they haven't done that well. Just to show you briefly what this looks like, if anybody's squeamish, don't look. But here I'm going to just show you a picture on the next slide of a wafer going into the human brain. It's the white one. And then on the next slide, you'll see a number of these. Usually they put 6 or 7 in and close up the brain. The next slide, if we could just put the next slide on. Actually, it's very hard to get good advice, but about 15 years ago, I was giving a lecture at MIT to chemical engineers, and my wife came, and I asked her at the end of the talk, what did she think? And she said, well, Bob, the talk was okay. That's actually very high praise. But she said, you had those 2 bloody slides on for about 10 minutes where you're going over all the details of the operation. She said, and Bob, all those chemical engineers were turning green. So ever after that, I've done what I did today. I showed it very quickly and then and gave a warning. Just to tell you briefly, there's a sequel to that. Is a couple of years after that, I gave a lecture, a dinner speech actually, to a group of neurosurgeons. And at the end, I did the same thing. I gave the warning. And at the end of the talk, a number of the neurosurgeons came up to me and they said, you know, those two bloody slides you showed? And I said, yes. They said, those were fine. You could have left those on as long as you wanted. They said, but those chemical formulas, to leave those on for a neurosurgeon, that's terrible. Anyhow, a lot of neurosurgeons have used these. This is now for an early study in Sweden and Finland, where they showed that you could get a 5-fold improvement in survival. And the next slide is a compendium of many of the clinical trials as of 2015 with 5,000 patients showing that and there's no cure. It prolongs life and relieves suffering. And now it's been approved for use for 24 years in over 30 different countries. And I think not only does it open the door to localized chemotherapy, which can be used in certain tumors, but even more so, it's opened the door to other areas, like our patents have been used, for example, in our technology for stents. The next slide's an example of that. Anybody that has heart disease, one of the most common treatments is to put a stent. It's like a Chinese finger puzzle that you put in a blood vessel when it's clogged, and it opens up the blood vessel. But about 50% of the time, you get what's called restenosis, and the blood vessel closes up. So now what they do is they take a different material polymer and they put a different chemotherapy drug, in this case Taxol, and they locally deliver it, and it prevents the clogging up. It prevents smooth muscle cell proliferation. In fact, Elazar Edelman, another one of my graduate students many years ago, and now a very well-known cardiologist, did a lot of the pioneering work on this, along with different companies like Boston Scientific and Johnson & Johnson. Well, one other thing that I want to go over tonight is not only can you use materials to help deliver drugs better, but you could also use it to create new tissues and organs. And here I've worked very closely with Jay Vacanti at Mass General and Children's Hospital. The next slide shows a little baby. This is from 1993 that he was treating, who was dying of liver failure. And the only way a child like that would live is someone else would die. And next slide. And Jay would, have to, get up in the middle of the night, do the transplant. But what he asked me, he said, well, maybe, Bob, we could make tissues and organs from scratch. What if we could take the patient's own cells, and now you could also take stem cells? Next slide is from a paper we wrote in Science. I'm sorry, if we could go back one. It's a paper we wrote in Science in 1993. It's been cited over 7,000 times, and it gets across the idea of what's now called tissue engineering. And the idea is you could have take the cells from the patient themselves or stem cells, and if you inject them into the body by themselves, not much happens. But cells are smart, and so we made these scaffolds, biodegradable scaffold, because the scaffold we want to go away after it provides the template for the tissue to form. We grow it in a bioreactor, and then you could make literally any tissue or organ. And there's lots of permutations of this that our students have worked on over the years to make virtually any tissue or organ. But just to show you a few things, the next slide shows you a scanning electron micrograph of polymer fibers with cells on them. And Prasad Shastri, one of my postdocs, now he runs an institute in Germany, he actually used techniques like 3D printing and CAD-CAM techniques to make virtually systems in virtually any shape. And the next slide shows just a picture of a nose. And here, what he did on the left-hand panel is you see a foam that he made in the shape of a nose. And you could literally make them in any shape, like you could have an upturned nose, whatever you want. And you could someday maybe take the patient's own cells to put on the nose. So anyhow, working with the Vacantis, we actually, and Gordana Vanyak and Lisa Fried, these have actually been put on animals, nude mice. And here you see on the next slide where on the bottom panel, and they don't reject, this has been put on to redone the skull. And the next slide If you look on the upper part, we've redone his cheek. And if you open the animals up, it's pure white glistening cartilage. Histologically looks like cartilage too. This is shown in the next slide. Now, the Army then at one point approached Linda Griffith, who was one of my postdocs, now professor at MIT, myself and Jay Vacanti, to see if we could make new body parts like ears for soldiers who came back from Iraq and Afghanistan. And Linda actually made a scaffold of the ear that's shown in the next slide. And you see that on the top of the ear, and on the bottom part, you see a scanning electron micrograph of polymer fibers, and they'll ultimately dissolve. But you also see cells and extracellular matrix here. And so you'll actually can make virtually any tissue or organ. And they put it on, just as a proof of principle, for the soldiers, they put it on a rabbit, and that's shown on the next slide, the human ear and the rabbit. And on the next slide, it's actually been done on patients. This is a 12-year-old boy that Jay Vacanti, who was my collaborator, treated. Noticed that he has no chest covering his heart. But like other 12-year-olds, he likes to play baseball. And so Jay actually took his own cells. We made a scaffold out of the polymer, put them on, and actually made him a new chest. And now he's 6 feet tall, and this was critical to enable him to live a normal life. We also have licensed this technology to a variety of companies who've made different tissues. I'll just pick one example, which is new skin, which is FDA approved, and new cartilage. Actually, I'll go over both of those briefly. Well, this is cartilage for skin. If we go to the next slide, what you see is a 2-year-old boy here. This is one of many, many patients who've been treated, but notice he's badly burned. But you can take the product, which are neonatal skin fibroblasts. This is shown in the next slide. You can actually cryopreserve that. You can put that in the child at the time of injury. That's shown in the next slide. And if you come back 3 weeks later, he looks like what you'll see on the next slide. And if you see, he's doing much, much better. And if you go to 6 months later, he's pretty much healed. And that's the next slide. And in fact, what this has all led to, and I'm just giving you a little bit about what we and some of my students and colleagues have done, are all kinds of tissues or organs. We're now, working on making new blood vessels. There's a company, one of my students started, Laura Nicholson, called Humacyte, that's in phase 3 to make new blood vessels. There's another company we started, and I should make a disclosure. All these companies I've helped and I've added equity in. But at any rate, we've had frequency therapeutics is using molecules that can stimulate cell growth for new hearing. In vivo therapeutics is working on spinal cord repair, and many, many people all over the world are working on this. The other big thing that it's led to are organs and tissues on a chip, which you could use to treat heart disease, or to study molecules that might treat heart disease or liver disease or other things like that. So we've been very excited about what's happened in this area. I thought I'd end the talk on one last slide. Obviously, in the era we're living in, with COVID, it's really critical to see what we can do. And what I've tried to do, in addition to the research that our laboratory's been doing over the last many years, is to apply some of the things to either through companies that we've started or in our lab itself to these kinds of problems. So, and I thought I would just, with this last slide, mention at a high level some of the things that we're doing. One that's gotten a lot of publicity is, again, as I mentioned, a company I helped start, which is Moderna. And here we've used nanoparticles to encapsulate messenger RNA to deliver, to hopefully come up with a vaccine to treat COVID. And this is in very advanced clinical trials. We've also worked with the Gates Foundation to come up with what we call self-boosting vaccines. Most of the time when you look at COVID and really anything, you have to keep giving, or for a lot of ones, you have to keep giving injections over the years. And actually in the developing world, which is why the Gates Foundation was so interested in it, is people just don't do it. So what we came up with, and we published this in Science, the journal, is a way to actually do a new type of printing technique where we can have bursts. We can put the vaccine in and we can get bursts not only at time 0, but at say 1 month, 5 months, 6 months, a year. This was work done by Anna Jakulenik and others in our lab. I also, one of the other big things, and this may seem mundane to people, is packaging. You know, there aren't even enough glass vials or other things for these vaccines. So here I've been helping SiO2 for many years. This is a company in Alabama come up with a new kind of packaging technique. You know, they just got a $163 billion BARDA grant to make these for COVID. I've also worked on, this is the 4th point, with David Edwards, one of my former students, and Denny Osiello, who used to be chief of medicine at Mass General, to come up with new nasal sprays that can hopefully combat COVID. And then we've done a lot of work with masks. James Byrne and Gio Traverso, who've worked with me, have come up with masks. And I also had the pleasure, as had other people in our lab, of working with New Balance, a sneaker company, to come help them design better masks. And they've made millions of masks already for healthcare workers and others. And then we've worked on split ventilators, on also Syed, who's a former guy in our lab, has worked on ultra cheap ventilators. And then we've, with Seventh Sense, a company I helped start, we came up with ways to do microneedle blood draws, which are essentially painless. So you can hopefully do serology tests. And finally, as I mentioned, developing organs on a chip. One of the ones that we're working on, we just got a grant with Liwei Shai to come up with a brain on a chip. And so we're looking at, we're starting to develop that and look at the neurologic impact of COVID on that. So anyhow, those are just some of the things that we're trying to do. And it's been a pleasure to speak to you tonight, and I'm happy to try to answer any questions. Thank you so much.
Thank you very much, Bob. That was terrific. I'm going to try to feed you some questions, and audience, please add questions to the Q&A, and we'll cover as many as we can in the 10 or so minutes that we have. I'm going to start with the technical questions, although there are some about how you mentor people, and I'll come back to those. But the first technical question is it possible to encapsulate different types of molecules in the same assembly, for example, so that cells might encounter factor number 1 first and then factor number 2 a little later, in order to coax cells down a particular path for various applications?
Yeah, well, the answer to that certainly yes, and there's different ways to do it. I mean, you can, one way, which actually Paula Hammond at MIT has done, is to actually use what are called layer by layer techniques, and so you can absolutely do that. So there's different ways that can be done, though that's a particularly attractive way, I think, the layer by layer technology.
Next one's about tissue engineering. Using scaffolding to grow tissues and potentially organs from a patient's cells would be life-changing for many people. Currently, hundreds of thousands of people are on waiting lists for donor organs, and transplant recipients must receive immunosuppressants. Would you speculate on the timeline when these will become more readily available for organs beyond skin?
Sure. Well, that's a very good question for which there's not a very good answer. I think it's hard to say. I mean, some are moving, quickly. I mentioned Laura Nicholason's work, where they're making new blood vessels, and I think that they've done some phase 3 trials, which is the last set of trials, which look very promising. But it's really hard to say. I mean, there's sort of two issues. One is science, and the other is funding. And these, as people know, it takes an enormous amount of funding to develop these things, and that makes it hard to predict on the timing. But I'm certainly optimistic that many years from now, there'll be a lot of examples of that. but it's really hard. I never want to give false hope. I mean, that's one thing that Judah Folkman also taught me, that you just don't want to tell people that something's going to be ready before it is, and it's really hard to know.
Yeah, I mean, I'm going to ask sort of my own question here. I mean, part of the problem is that these things really can work in a kind of a individual patient kind of thing, but it's hard for a company to make money manufacturing these things because it's expensive and the manufacturing processes are difficult. Is that true in your view?
I think it is true. I think that gets to some of the issues that come up, when people are so concerned about pricing, and I certainly understand that. But you're right. If when you make it so hard for companies to make money, then they're reluctant to do it. There are some examples where companies have made significant money in this area, but it is an expensive technology. All drug development, and tissue development even more so, I think, is expensive. And so it's so your question is a very, very good one.
Yeah, I mentioned in my introduction about your work in diabetes, and there's a question sort of asking about what kinds of things you're doing, if any, or what you think sort of the engineering prospects are for addressing type 1 diabetes these days.
Yeah, well, we're actually doing a lot in diabetes. I'll give two kinds of examples. One really came because the Juvenile Diabetes Foundation came to see me about 12 years ago, and they said, could we make an artificial pancreas by encapsulating islet cells in certain polymers? And, that was an idea, of course, that had been advanced before, but whenever people created the polymers, they'd always get encapsulated with fibrous tissue. That's just been a classic problem. And they said, well, could we solve it? So Dan Anderson was one of my postdocs then. He's now a full professor at MIT. And he and I had this idea that maybe we could develop new kinds of chemistries and high throughput screening techniques to synthesize literally thousands of polymers and hopefully find some that might work. And actually, for the first 7 or 8 years, we didn't publish anything, but the Juvenile Diabetes Foundation was great, and they kept funding us. And in the last 4 or 5 years, we've published actually 9 papers in different Nature journals, really elucidating the mechanisms of biocompatibility, taking them into non-human primates, and really getting no encapsulation in non-human primates. So we started a company, Sigalon, and they've really advanced this quite far, and they actually have gotten Eli Lilly gave them $500 million. I mean, 80 up front, but 500 overall to try to bring this to fruition. So I'm hopeful, the COVID thing notwithstanding, that clinical trials might start within a year on that. That's one example. The other example I wanted to give is we've also worked closely with Novo Nordisk, and we've actually come up with a way. We published this in Science last year with Gio Traverso and others, that we came up with a pill that you can swallow, that children could someday swallow, that could deliver insulin. So you could have oral insulin. And it's kind of a certain way of having a little pill that's almost like a Weeble Wobble toy that you can swallow. But then when it gets in the stomach, it Weeble Wobbles the right way and injects insulin right into the stomach and doesn't hurt. And and they've been great to work with. So those are a couple of examples in diabetes that we've been working on.
There's a lot of questions about your own professional style and things that you've encountered in your career. So I'll just try a couple of those. With all the different rejections that you've received over your career, how did you not give up on science, and what advice would you give to scientists trying to tackle risky or difficult problems?
Well, Matt, and I'm sure everybody has struggles whenever we go into science. People don't like your grants, and people think if your ideas are too futuristic and this won't work and that won't work. To me, it's like you have to just keep your head down. And when I was a young guy, I was discouraged. It would be wrong for me to say that. I got depressed. I was very sad about getting rejected. but I believed in it. I saw with my own eyes that these things were working, and I felt like I wasn't gonna give up because of that. But so it's discouraging, but you have to just keep trying. And same thing with grants. I mean, I just I thought I was right, and so I kept trying. And I don't know. It's a lot of it's just persistence, and in science, I think it's tough. I look back at my career, I've probably failed many, many more times than I've succeeded. But, it's like a couple of successes, like I can give a talk tonight on, there's 45 years of work. And I mean, I certainly mentioned things where we failed, but, a couple of successes and, people think you're okay. So that's,
Yeah. As a person to whom you've been compared once said, Thomas Edison, I've never failed. I've just discovered thousands of things that don't work.
Yeah, well, I think I could relate to that.
Yeah. Another kind of personal question, or at least your own psychological, as a principal investigator, do you consider yourself, or do you think that ideas that are more curiosity driven or more application driven are more powerful? I mean, how would you say you've been motivated, by curiosity or by applications? I'm sure applications have figured prominently. So where has curiosity figured into your own career?
Well, really both. It's like, I mean, curiosity, of course, is I think what helps me and others make these findings in the first place. But I guess what I'd say is what's been important to me, and maybe it's being an engineer like you are, is so you can be curious and you can discover something or invent something. But that's just, to me, the first step. I mean, I guess when I look back at my life, I really wanted to not just write a paper, and we've written a few, but I wanted to see the curiosity driven discoveries and inventions make a difference in people's lives. And maybe that comes from the school that I helped start a long time ago and seeing people feel satisfied. Maybe it comes from working in the hospital as well as being an engineer. I'm not sure, but really, to me, it's both. I mean, I really feel that both those things are incredibly important. I don't make even a distinction. I've told people when they come to the lab that the important thing is to make a contribution. And it's whatever contribution you feel you can make, whether it's a curiosity-driven discovery or something that you can take something like that and apply it and make the world a better place. And both are good.
We're a little bit over time, but I'm going to ask one more question that relates to you talked about how Judah Folkman, was took a risk on you and mentored you, and now you've mentored many successful and influential scientists. What kind of advice do you try to impart to them? And another question along the same line is, what do you look for when you think about taking on the responsibility of advising or mentoring someone?
Yeah, well, let me do the second one first. And the second one to me is I basically look for not people who are, let's say you get a grant to do X, whatever X is, and people might say, well, you should really get somebody who's got a certain type of background. I don't look for that. I look for whoever I'd say is the best athlete overall, whoever I think is just going to be terrific. And that, and even if they don't have a lot of experience, the fact that their advisors tell me they're the best person they've ever seen, and that they've done just great work. That's kind of what I look for, is just somebody who I think, can change the world. I think in terms of advice, I think if I learned anything from Judah Folkman, it was to believe that almost anything was possible, to recognize that you're going to have a lot of people criticizing you and not believing in you and telling you that you're wrong. So don't give up. So I think it's those two things, that try to think of ideas that can make a real difference in the world, and recognize that you're gonna run into obstacles, but and maybe a lot of obstacles, but don't give up.
Thank you very much, Bob. We've covered most of the questions, and it's been a pleasure to to hear you and interact with you again over this hour, as we have many times. And thank you for taking the time to give this lecture to the distinguished audience of the Friday evening lectures at the Marine Biological Laboratory. Thanks again.
Well, thank you. It's really been an honor and a pleasure. And Matt, it's always a pleasure to interact with you and also to see the amazing job you've done at University of Chicago and elsewhere. So anyhow, it's been a pleasure for me. Thank you.
Thank you, Bob.